Slide 1. Peripheral circulation is a series of slide tape programs developed and produced by the American Physiological Society. This is Exchange 1, the Capillaries and Solute Diffusion. Content experts, Dr. Brian R. Duling and Dr. Paul C. Johnson. 2. The earlier programs in this series present discussions of hemodynamics and the arterial and venous systems. Later programs discuss the local and neural mechanisms that control systemic circulatory function. Please note that a printed study guide accompanies this program. It contains the full program text and references for additional study. 3. In hemodynamics, we analyzed the viscous and inertial resistance factors responsible for the pressure drop observed in the circulatory system. 4. In Part 1 of arterial and venous systems, we discussed the organization of the vasculature and the distribution of vessel size, flow rate, and resistance throughout the network. 5. We also described the structural components of the vessel walls, the elastic properties of the vasculature, capacitance, and the force balance between transmural pressure and circumferential stress. 6. In Part 2, we analyzed arterial systolic and diastolic pressures, the determinants of pulse pressure and mean arterial pressure, the transmission of the arterial pressure pulse wave, and venous blood pressure, storage, and return. 7. This program discusses the anatomy of the terminal vessels of the microcirculation and the endothelial histology of capillaries relevant to the exchange of substances between blood and tissue. It also describes the process of diffusion of solutes across the capillary wall. 8. Your first objective is to be able to identify and sketch the detailed anatomy of a generalized microvascular bed. 9. The terminal vessels of the microcirculation consist of the arterioles, capillaries, and venules. In the broadest terms, the arterioles are responsible for flow control. The capillaries are responsible for exchange between blood and tissue, and the venules act as collecting vessels and volume reservoirs. 10. Recall that vessel diameter and wall thickness decrease progressively down the arterial tree. At about 50 microns in diameter, arterial vessels lose all but one layer of smooth muscle. These vessels are called arterioles. The muscle cells provide the means for physiological control of arteriolar dimensions and flow control. 11. Those arterioles feeding directly into capillaries are called terminal arterioles. They are about 10 microns in diameter. The entrance to the capillaries in some tissues is surrounded by a smooth muscle cell that can regulate flow into individual capillaries. This cell is called the precapillary sphincter. 12. The exchange of materials between the circulatory system and the cells of the body takes place mainly in the capillaries. These vessels are small, averaging only 4 to 8 microns in diameter. They are approximately 500 microns in length. 13. In some tissues, there is a second category of small vessels connecting the arterioles and the venules. These are the thoroughfare channels. They are only slightly larger than capillaries, 10 to 15 microns in diameter, but they differ from capillaries in that they have a smooth muscle media over about 1/2 their length. Capillaries can originate from these thoroughfare channels. 14. On the venous side of the circulation, the capillaries coalesce into small venules, which are simple endothelial tubes without smooth muscle. These combine with other venules, and at approximately 30 to 80 microns in diameter, a smooth muscle coat appears on the venous side of the circulation. 15. As a rule, what element of vascular geometry is varied to control flow? What vessel type is most important in flow control? Please stop the tape while you answer these questions and those that follow throughout the program. (16) Vessel radius is varied to control flow. Terminal arterioles are the most important vessels in flow control. In some tissues, flow to individual capillaries can be controlled by closing or opening the precapillary sphincter. 17. Make a sketch of a generalized microvascular bed. Indicate vessel diameters and the presence or absence of smooth muscle cells. 18. Your sketch should show the following. Arteriolar diameter ranges from 50 microns down to 10 microns in terminal arterioles. Arterioles have smooth muscle along their entire length. 1/2 of the length of a thoroughfare channel is covered with smooth muscle. There is no smooth muscle on a capillary. The first venules with smooth muscle coats have a diameter of 30 to 80 microns. 19. Your next objective is to be able to sketch and name the elements of endothelial histology relevant to the transfer of materials between capillaries and tissues. 20. 3 different processes are involved in the transport of materials across capillary walls: diffusion, micropinocytosis, and filtration absorption. Broadly speaking, diffusion is responsible for transport of small and large solute molecules. Micropinocytosis is responsible for large solute molecule transport, and filtration absorption is responsible for fluid distribution between the vascular compartment and the interstitial space. These 3 processes operate in concert and interact with one another in determining the transport of materials between blood and interstitial space. For example, water can move by diffusive transport, and solute molecules are carried in fluids during filtration absorption. For the purposes of this program, however, the 3 processes are treated as separate events. 21. Understanding capillary exchange requires a rather detailed knowledge of the anatomy of the capillary and the histology of the capillary endothelium. The capillary is basically a simple tube formed of overlapping endothelial cells. 22. Capillaries are lined with a thin non-structural protein coat. The protein coat may play a role in establishing the permeability of the capillary, especially to proteins. The capillary is covered by the basement membrane. In most tissues, the basement membrane is not a major factor in determining exchange. 23. The capillary endothelial cells are complex structures. They are separated by intercellular spaces which are 50 to 100 angstroms wide. Small materials can move across the capillary endothelium through these aqueous channels. These spaces are reduced at intervals by gap junctions and in some capillaries by tight junctions. In the following slides, we describe 3 degrees of endothelial continuity. 24. First, a continuous endothelium in which there is close apposition of the endothelial cells exists in the lungs, in striated muscle, and in the central nervous system. 25. A second type of endothelium possesses marked regional thinnings of the capillary wall. These are called fenestrations. Some of the fenestrations are open and some are closed. The degree of fenestration is highly dependent upon the type of tissue. Fenestrated capillaries are found in the renal glomerulus and in the intestinal mucosa. 26. The third type, discontinuous endothelium, has wide spaces between adjacent endothelial cells. Discontinuous endothelium is found in the spleen, in the liver, and in bone marrow. 27. The last aspect of endothelial histology that must be appreciated is the presence of micropinocytotic or plasmalemmal vesicles, which are distributed widely in the endothelial cytoplasm. These vesicles are formed at the cell surface by in-pouchings of the cell membrane. During this process, the vesicles pick up small amounts of fluid and solutes. The vesicles close, bud off from the cell membrane, and then move by Brownian motion across the cell. Some of these vesicles open to eject their contents into the pericapillary space. The full importance of this process is not known, but it is probably responsible for a large fraction of the transcapillary exchange of very large molecules such as globulins. 28. On occasion, the endothelium may be perforated by the fusion of two or more vesicles. The resulting vesicular chain may provide an additional pathway for transcapillary movement. We have now mentioned two pathways by which lipid-insoluble substances may diffuse across the endothelium, through intercellular spaces and through fused vesicles. When these two pathways are used, the moving substance traverses the capillary wall without entering the cytoplasm of the endothelial cells. 29. All aqueous channels in the capillary wall are referred to by the general term pores. These include intercellular spaces and chains of fused vesicles. In addition to these, there are very small pores that penetrate the endothelial cell membrane. These pores, which provide aqueous routes into and out of the cell's cytoplasm, are only a few angstroms in diameter. 30. Sketch and label a cross-sectional view of continuous capillary endothelium. 31. Your sketch should include endothelial cells, the protein coat and basement membrane, intercellular spaces, vesicles, and endothelial membrane pores. 32. Name the two types of channels that traverse the endothelium and provide possible routes for transcellular movement of lipid-insoluble substances. Name the three types of capillary endothelium, and name one type of tissue in which each type of endothelium is found. 33. The two types of channels that traverse the endothelium and provide possible routes for transcellular movement of lipid-insoluble substances are intercellular spaces and fused micropinocytotic vesicles. Continuous endothelium may be found in striated muscle, lung tissue, and the central nervous system. Discontinuous endothelium may be found in the spleen, liver, and bone marrow. Fenestrated endothelium is found in the renal glomerulus and intestinal mucosa. 34. Your next objective is to be able to describe the mathematical terms of importance in Fick's law of diffusion. 35. Not all exchange takes place through pores in the endothelium. Lipid-soluble solutes do not require pores. They can dissolve in the membrane lipid bilayers and traverse the cells via the cytoplasm. Diffusion through the cell membranes is the means by which virtually all the oxygen is supplied for oxidative metabolism, and the process by which carbon dioxide is removed from the tissues. In contrast, small lipid-insoluble solutes, such as glucose and lactate, must traverse the endothelium via the aqueous pores. 36. Diffusion across the capillaries is fundamentally no different from diffusion under any other set of circumstances. It can be described as the random movement of individual molecules down a concentration gradient. The magnitude of the diffusion rate, or diffusive flux, can be predicted to a first approximation by Fick's law of diffusion, which states that the diffusion rate is equal to the product of the concentration gradient, the area available for diffusion, the reciprocal of the diffusion distance, and the diffusion coefficient. 37. In this example, a difference in solute concentration exists between the blood, C in, and a localized area of tissue supplied by the capillary, C out. The diffusion rate, N, is directly proportional to the concentration difference, delta C. 38. The magnitude of the diffusive flux is also directly proportional to the surface area available for diffusion. For lipid-soluble substances, this is the entire internal surface area of the capillary. A equals 2 pi RL. For lipid-insoluble solutes, the relevant area is that occupied by all the aqueous channels in the endothelium. In both cases, the surface area for diffusion is usually controlled by changing the number of capillaries perfused. 39. The distance across which the substance must diffuse is also an important determinant of the diffusive flux magnitude. The total solute flux varies with the reciprocal of this diffusion distance, delta X. For lipid-insoluble substances, the length of the aqueous channels is more important than the distance to a particular site in the tissue. That is, diffusion is restricted essentially by the capillary membrane. The resistance to diffusion posed by the tissue may be neglected. 40. The variables describing diffusion are linked by an empirically determined constant, the diffusion coefficient, D, which is characteristic of the diffusing molecule, the capillary, and the medium. To summarize, the transcapillary diffusion rate, N, equals the product of the diffusion coefficient, D, the area for diffusion, A, and the ratio of the concentration gradient and diffusion distance, delta C to delta X. The negative sign is used to denote the fact that the flux is down a concentration gradient. 41. With regard to diffusion, there are purely geometric advantages to the very small size of capillaries. First, since the ratio of a vessel's surface area to its volume is dependent only on its radius, vessels with a very small radius have a large surface area to volume ratio. Second, the average diffusion distance is reduced by a system of numerous small capillaries. The large surface area of a tissue's capillaries and the short diffusion distances facilitate exchange between blood and tissues, and the small volume of the capillaries allows the bulk of a tissue to be occupied by functional cells rather than by blood vessels. 42. Write the Fick equation and name the variables in the equation. Diffusion is the major transport process for moving what type of molecules across the capillaries? 43. N equals negative dA delta C over delta X. N is the diffusion rate, D is the diffusion coefficient, A is the area available for exchange, delta C is the concentration difference, and delta X is the diffusion distance. Diffusion is the major process by which small solutes, both lipid-soluble and lipid-insoluble, enter and leave the tissue. 44. Your last objective is to describe the ways in which flow rate and geometric factors determine the transcapillary diffusion rate. 45. The concentration gradient, one of the major variables determining diffusion rate, is a complex factor in capillary exchange problems. This is because the solute concentration in the capillary and in the tissue may vary along the length of the capillary. The degree of variation depends upon the rate at which a substance is delivered to the capillary and the ease with which it crosses the capillary. 46. For a substance that moves rapidly between blood and tissue, oxygen for example, the capillary concentration falls all along the length of the capillary. The tissue consumes from 20 to 90% of the oxygen that enters the capillary with the blood. 47. In determining diffusion rate, the relevant concentration is the average concentration along the length of the capillary. 48. For substances whose concentration falls along the length of the capillary, the exchange rate is dependent on the flow rate. When the flow rate is high, there is a greater supply of the diffusing material, so the intravascular concentration doesn't fall as rapidly along the capillary. When the flow rate is low, the supply of diffusing material diminishes all along the capillary. 49. This graph shows how intravascular concentration changes in high and low flow states. For example, if the concentration all along the capillary remains higher due to high flow, it follows that the mean concentration of oxygen in the capillary is raised. A higher concentration inside means that there is a larger gradient to drive oxygen diffusion into the tissues. 50. A substance whose diffusion is altered by changes in blood flow rate is said to be flow limited. Here, the diffusion rate is plotted against blood flow rate. Note that when very high flows are reached, capillary diffusion is only slightly increased by further increases in flow rate. 51. For substances such as proteins which move slowly across the capillary wall relative to their input rate, the intravascular concentration does not change significantly along the capillary. Therefore, the exchange rate is not dependent on flow rate. In this case, the exchange is said to be diffusion limited. 52. We have seen how concentration may influence capillary diffusion. The second major variable determining the transcapillary diffusion rate is the number of flowing capillaries. Capillary exchange is controlled mainly by regulating this factor. In many tissues, between 50 and 80% of the capillaries are unperfused in the normal resting state. When additional capillaries are opened to flow, the surface area for diffusion increases and the average diffusion distance decreases. Both changes increase the overall rate of diffusion. 53. During times of increased demand for nutrients, closed capillaries can be opened by either relaxation of terminal arterioles or by relaxation of precapillary sphincters. In either case, the capillary exchange area is increased by increasing the number of perfused capillaries, not by increasing the size of patent capillaries. 54. As stated earlier, the total surface area of the capillary can be utilized only by molecules that can penetrate the lipid bilayer membrane of the endothelial cells. That is, by molecules that have a relatively high lipid solubility. The respiratory gases, carbon dioxide and oxygen, are both adequately soluble in the membrane to permit them to diffuse across the entire area of the capillary endothelium. 55. For lipid-insoluble substances, however, the area available for exchange is not the total surface area of the capillary. Charged lipid-insoluble substances, such as sodium, which cannot easily penetrate the cell membrane, are restricted to passage through the aqueous channels that traverse the capillary wall. Recall that two types of channels are available: intercellular spaces and fused micropinocytotic vesicles. In striated muscle, only about 1/100th% of the total capillary surface area appears to be occupied by aqueous pathways. 56. The diffusion coefficient is the third parameter determining the rate of transcapillary diffusion. For lipid-insoluble substances, this is a complex parameter because the aqueous channels and some of the diffusing molecules may be on the same order of size. For example, the principal transcellular aqueous routes through the endothelium of skeletal muscle are 50 to 100 angstroms in diameter, and albumin has a molecular diameter of 72 angstroms. For these molecules, the diffusion coefficients are less than would be predicted from observing free diffusion in solution. 57. The size of many of the molecules of biological interest approaches the size of the aqueous channels, and diffusion is restricted. This restricted diffusion rate becomes very low for large molecules. Notice that the rate for very large molecules is not zero, however. To explain this, it has been proposed that the capillaries are either penetrated by a small number of very large pores, or that micropinocytosis carries the large molecules across the endothelium. Both factors are probably important. 58. To summarize, transcapillary diffusion is a complex problem. For a lipid-insoluble substance, the diffusion coefficient, D, depends on the relative sizes of the diffusing substance and the pores. The diffusion area, A, depends on the number of capillaries perfused and the lipid solubility of the diffusing substance. The concentration gradient, delta C, may vary along the length of a capillary, depending on the rate at which the substance is delivered to the capillary and the ease with which it crosses the endothelium. Experimentally, it is possible in many cases to determine the magnitude of the ratio between the diffusion coefficient, D, and the diffusion distance, delta X, even though the absolute values of the two components are not known. In order to simplify the problem, the diffusion coefficient and the diffusion distance are combined into a single term, permeability, P. 59. How is the rate of transcapillary diffusion controlled? Name the anatomical structures responsible for this control. 60. The rate of capillary diffusion is controlled mainly by opening and closing capillaries, which alters the surface area available for exchange and the diffusion distance. Control of capillaries is accomplished by the terminal arterioles and the precapillary sphincters. 61. What portion of the total area of the capillary endothelium is potentially available for transcapillary diffusion of a lipid-insoluble molecule such as potassium? A lipid-soluble molecule such as xenon? 62. Potassium diffusion is restricted to aqueous channels. A molecule such as xenon, which is highly soluble in lipids, can diffuse through the entire capillary surface. 63. Define the permeability coefficient. When flow through a capillary is increased, how does it influence the solute concentration in the plasma within the capillary? Define a flow-limited substance. 64. The permeability coefficient is the ratio of the diffusion coefficient to the diffusion distance. When flow is increased, the concentration of the solute in the plasma is increased. A flow-limited substance is one whose capillary exchange rate is influenced by the blood flow rate through the capillary. The dependency on flow arises from the fall in concentration of the substance along the length of the capillary. 65. To summarize, we have seen that diffusion is the dominant process by which solute molecules must move from the capillaries into the tissues, and by which metabolic waste products must be removed from the tissues. We described in detail the determinants of the diffusive flux for solutes. 66. We mentioned earlier that water can move by diffusive transport. Water can also flow as a fluid through channels in response to a pressure gradient, as when water flows through a pipe. Water, carrying its dissolved low molecular weight solutes, may flow from the capillaries into the tissues, which is called filtration, or from the tissues into the capillaries, which is called absorption. The balance between filtration and absorption is the major factor determining the distribution of body fluids between blood and tissues. These concepts are discussed in detail in the next program, Exchange 2, Filtration and Absorption of Fluids. 67. This concludes Exchange 1, the Capillaries and Solute Diffusion. To review the concepts that have been presented, and for a list of additional readings, please refer to your study guide. This program is part of the Peripheral Circulation Series. It was developed and produced by the American Physiological Society. Copyright 1981.